This is a production of Cornell University.
So what's a herbarium? An herbarium, I don't know, British, whatever you want. It's a place where we keep dried plant specimens. Now I'm going to go through everything very quickly here. And it's not as funny as most of my talks. I just want you guys to know that. It also includes liquid preserved specimens. Mostly, most herbaria have some, but I think we're kind of moving away from that because it's a pain. Oh, do I need the microphone? Can you guys hear me? No? All right, sorry about that. Can you hear me now? No? There, yes. I have to still screen. Okay, good. All right. So there are other things that are included in herbaria, typically archive field notebooks, associated literature, reprints. When I first came here, we had a massive reprint collection. We sent most of that off to various places, and now they wish they didn't have it. Early herbaria were often established primarily for the study of medicinal plants, and I'll go ahead and move on here. So this is the first real herbarium. It was by Luca Ghini, an Italian physician. Again, most herbaria were connected to medical enterprises, really, pharmacists, medical doctors, and so forth. So we'll move along without much ado here. But early herbaria oftentimes were also bound into books. So this is an example of one from 1603 from the British Museum. I just got that online. And then this is one of ours, which is as late as, this is for around maybe 1915, of plants from our area here. Now, now we put specimens on herbarium sheets. I mean, this has been going on for a long, long time. And this is just an example of some of our documentation of the Geneva experiment station there. Those are three early varieties of grape, 27, 23, 3. And how do we store our specimens? If you guys come to the open house, you'll see this. We put our specimens into these big cabinets, which are insect proof. Insects are one of the real problems we have in maintaining herbaria because there are certain kinds of little beetles and things that love to eat herbarium specimens. There's even a species of beetle that likes to eat the glue that we glue them on the sheets with. Now, I can't go on without mentioning some of the other collections that are here at Cornell, including the plant pathology herbarium. That's it. I'm not talking about it anymore. The Cornell University Museum of Vertebrates, insect collections. Oh, I forgot the brain collection. Oh, well. Okay, so what about us? We have about 950,000 plus specimens. We're not sure. It could be as much as 1 million. It's really, until we get it all digitized, database, we won't know for sure. But that's an estimate. I think it's probably closer to 1 million now because we've been saying 950 for a long time. There are about 250,000 of those specimens have been digitized. And all of our New York specimens have been digitized, the wild ones, not all the cultivated specimens. This is Anna and Peter, who are our employees, our staff at the herbarium. And I want to give them a shout out right now because without them, we couldn't have even done this presentation. There's a lot of materials here that they helped assemble, produce slides and so forth. Speaking of which, our herbarium actually represents the fusion of 3 historical herbaria. The CU Botany Herbarium, the Wiegand Herbarium, and then Bailey's Herbarium. And we're not going to go into detail here, but you can, we've now taken this name for the entire herbarium. So we refer to the herbarium as the Bailey Hortorium or BH Herbarium, and that's the official acronym in Index Herbariorum, but you should remember that it is, there are other herbaria that have been fused into it. What are some of the strengths of our herbarium? Each herbarium in the world has different strengths. In other words, different sets of specimens that are they have a lot of, or typically this is because some worker has been there that was interested in that group. And some of ours are cultivated plants. We have about 250,000 specimens of cultivated plants, and this is because of the legacy of L.H. Bailey. We have, of course, a lot of New York plants, about 110,000 specimens, New York State plants. We also have an incredible palm collection. There are two really good palm collections in the US, the New York Botanical Garden and the Bailey Hortorium. And we have, and I don't have a number there. We forgot to get the number. I apologize. Anna, how many do we have? Bob, nobody knows how many palms we have. Okay, we have a lot. The problem with palms, which you'll see in a moment, is they're really difficult to curate, store, and all that because they're so bulky. We also have a premier collection of Fagaceae, mostly oaks, and that came through me. C.H. Muller, who I worked with, gave us his collection. So we have a total of about 15,000 specimens of oaks, which are mostly from Mexico and Central America and the Western US. But we also have an excellent complement of our New York and Northeastern oaks that were already here before we got Muller's collection. And because of Muller, we have a lot of type specimens from oaks of Mexico and Central America. We also have a very strong collection of rubus, raspberries, blackberries, so forth and so on. Bailey was a rubicologist, okay? And we also have a very good collection of Cyperaceae. There are other collections we have that are really good, but these, I just kind of hit some of the high points here. All right, so why are the palms a pain? Here's how we have to store the palms. Mostly, well, there are some palms we do put on regular herbarium sheets, but a large number of the palms are very bulky in the collections and they go into boxes. They take up a lot of space and it's difficult to photograph them if you it's a whole table to photograph one specimen and so forth. All right. We also have a very strong bryophyte collection, which I almost never look at. Right. However, we do have Jonathan Shaw, who's now an adjunct here. And pardon? About to be. Oh, he's not an adjunct yet. Think he's going to make it? All right. Sorry. But anyway, Jonathan has connections here, and he's been working quite a bit now in our herbarium on the sphagnum collection. Now, you see, we have the opposite of the palms in terms of curational issues with mosses. Mostly mosses are put into envelopes, whatever, packets, and sometimes we have as many as 8, 10, or more on one sheet. And sometimes you'll see them actually in boxes like that. So they're a pain in that. Okay. We have a pretty good seed collection. It's not huge, but it is a nice thing to reference if you're trying to figure out, say, you have a bale of hay and what's in the hay or something like that. You don't have to go search through the herbarium. You can go look directly at some of the seeds. So what's the traditional importance of herbaria? Mostly in the old days, herbaria were used pretty much by plant taxonomists. We have plant identification, floras and monographs, vouchers, whereby, say, I do an anatomical study. I take a specimen and put it in the herbarium and mark it as used in that study. It's documentation. It's a huge part of making science repeatable. Herbaria are a very, very important part of that, is particularly in the older literature. Type collections, which are species name references. You can get species distributions, like for a flora or something like that. And then there are a lot of other things that you can get out, traditionally, out of a herbarium. We image, we're in the process of imaging our specimens. We have about 200 and, oh, I thought the number was up there. It'll be in a different place. We have about 220,000 specimens imaged. Now, I'll admit, it's my fault. These aren't online yet. They'll be coming online in the next 3 to 6 months. But I will show you the preview of what they will be when they're online. Now, I want to go on and talk about herbaria in terms of more modern uses. And of course, DNA is everything now, right? It's not DNA. You guys don't care if I didn't put DNA or molecules in the title, you wouldn't be here. So basically, herbaria can be viewed as a giant phenotype DNA archive. All right. So if you look at this paper that was done in 2020, Lenka, who I know from the Czech Republic, they did some studies and they found out that the specimens as old as 200 years, you can extract pretty good DNA out of. And it's not how old they are, it's how they were pressed and the condition they were in if it was fresh, okay? So Dan, I don't have the numbers up here, but Dan has, within our herbarium, he's only, how long been here, Dan? 2-3 years? Yeah, and you've already have 67 specimens, I think, you've extracted DNA from. Didn't you send me that? No, but I mean, it's in our herbarium. You sent me the number 67. Okay, let's go on. Doesn't matter. Now, 67,000? No. Okay, anyway, so if we look at this as an archive, what is that up there? You can put the X. It's not on my screen, it's why I didn't see it. Just where's the mouse?
It's right down on the, in the middle.
On the mall, actually.
It's on the word vascular.
It's on the word vascular. Where's the word vascular?
You just move the cursor to the.
Okay, you do it. I can't do it on my own computer. I mean, it's possible. Got it? Sure. Yeah, see, you can't do it either. Sure. All right, we got to move on. Okay, it's not, it's still there. No. You did it last time.
I know.
All right. You did it for Jonathan, actually, right? Yeah.
There it is.
But look, while we're doing this, let's talk about this. So worldwide, and this number is more or less correct. I think if it was in the abstract, it's probably wrong. There's 397,000 estimated herbarium specimens in the world.
Million.
Million. Did I say thousand? I meant million. Okay. 397 million herbarium specimens in the world. Now that's just specimens. And I'll show you in a moment. Many of these specimens have more than one plant on. So the number of potential DNA samples there is much higher. Now, you can take a herbarium specimen and nobody uses a PCR anymore. I'm aware of that, but you can get DNA out of it, right? Many times, not always. So as an archive or a place to keep DNA with an associated phenotype is directly connected. There's nothing that's cheaper, greener, more permanent, and has the phenotype. It's hard to imagine keeping your DNA samples in the freezer for 200 years. I think that would be a little bit too expensive. Now, silica is a good thing, right? So probably I'll talk about a little bit at the end about how we might modify what we put into the herbarium. So we'll talk about that. Now, one of the things that hasn't been exploited, as far as I know, there may be a paper on this or not, but if you can get DNA out of these specimens, we actually have little population samples, oftentimes, on these specimens. So that's another aspect of it that might be of interest. So let's go on to the metadata, right? So that's DNA in the herbarium. Everybody kind of knows about that. We also have metadata-based research in the herbarium. So for instance, herbarium labels have various kinds of information on them. We can geo-reference those labels to get localities. Like that's what we mostly would be interested in from GBIF, Tropicos, iBigBio. NIFA is the New York database. And the applications of the metadata are varied, but the most important one right now for global climate change is probably niche modeling, SDM generation, species distribution models, okay? We can also, though, we can do species mapping, we can do phenological studies, we can track invasive plant introductions, looking at the metadata, and we can also look at rare and endangered and extinct plant distributions. Here's a paper that was done about extended specimens using those to study global climate change, and you see there are a whole range of possibilities that you could use herbarium specimens for that have relevance in the study of global environmental change. We don't have time to go into great detail there. Now, these other uses that are emerging, again, I already mentioned most of these. These are just some of the papers that have recently been done that use herbarium specimens. A newer thing that's happening is to do isotope analysis of specimens, to look back in time to see, for instance, what the CO2 levels were as captured by the plants. There's also, of course, phenological studies. By that, I mean that you can go back through the herbarium and see if flowering times have shifted over time. There have been several papers like that. And of course, the idea is that flowering times might get earlier if the climate is warming. I had a student that graduated a few years ago, and we kind of reversed this idea. The idea for Rob's dissertation was to take the distributions of the plants and estimate the climate at that site. So you go to a site, let's say you have 100 plants growing there, you can do a likelihood analysis to say, based upon the envelope of climate that you typically find those plants in, you can estimate what the climate is at that spot. And that can be good for fossil work if you're not going back too far in various other applications. We can also use herbarium specimens to track invasive plants, okay? For instance, we can find the earliest records of an invasive plant in a particular area. And if you do that across the whole continent, then you can kind of track how an invasive plant might have been introduced and how it spread, when it was here, when it got to New York State, when it got to Tompkins County, things like that. That's, of course, assuming you have enough herbarium specimens, which you guys need to go out and get as many as you can, and so that we can do this in the future. So here's purple loosestrife, 1875, swallowwort, 1917, and garlic, 1878. So this is an incredible resource for understanding how invasive plants have moved. Now, we also, of course, we have genetic data with these. In theory, if we can extract the DNA out of them, we can also look at origins. Which part of Europe did this come from, or something like that? A lot of these invasives were due to very limited introductions. So we can tell more or less how they came, where they came from. And this is a very good way to do that. We also have extinct plants in the herbarium. These are 4 examples that we happen to have. We probably have a few more. What can we do with a herbarium specimen of an extinct plant? Well, of course, it's definitely a record of one of the last times that it was seen, if it's late in the, near when it went extinct. But we also have DNA that we might be able to extract from these. We can look at the morphology, which we no longer have, things like that. Now, I'll just point out that there's a new kind of jargon that's being used about this very thing called extended specimens, where you have a specimen and then you have other things that are associated with that specimen. So an extended specimen network can have all kinds of things in it. Of course, you have the images. You can look at the actual image of the specimen as an extended aspect of it. Then you have data that you get off the labels, like the metadata, so forth and so on. And then you can connect these things together in your databases. I want to briefly talk about our projects and collaborations. This is a slide that Chelsea shared with me. She forgot to put her name on it, so I stuck it there, the spec lab. Okay, so they're doing all kinds of things that have either a direct connection to the herbarium or an indirect connection to the herbarium. Now, some of these things, they're coming out of large databases of herbarium data, and some might be working in a particular herbarium like that. I'll leave that up for a moment. I'm not going to go over it all. So just soak it all in. Here's a slide that we put together from Joyce, who's no longer here. She said, that sounds terrible, doesn't it? She's at NYU. no longer with us. Okay, so. But she worked on evolution of vines, vine habits, the anatomy, and so forth. So not only can she document what she's doing in the herbarium with a voucher specimen, but you can actually take specimens that have been there for a long time and get plant anatomy out of those specimens, right? You can take sections of the stems. So if you're interested, oh, well, does this family or this plant have a similar anatomy? Maybe it hasn't been looked at. You can get it out of the herbarium. I didn't have a picture of Josh. Sorry, Josh. Where's Josh? Yeah, okay. So, but Josh is doing a project in the herbarium. It's a rotation under myself and Alejandro Gandolfo on the trichomes of the Berneliaceae. Now, these trichomes, as many of you know, have adaptive significance in that they're able to take up water into the plant through the trichomes that are on the leaves and so forth. So Josh is looking at the distribution of these and the variation in the morphology. Anything else? That's good enough. Okay. So BH also has an ethnobotanical collection, and we're going to be soon adding another big ethnobotanical collection. It's a 16-foot trailer of a Krikorian. How do you pronounce his name? Krikorian. I always want to say Krikorian. Krikorian, who passed away recently, but he collected ethnobotanical artifacts in various places around the world, has some beautiful stuff, and he's giving it to us. All we have to do is find a place to put it. Maybe in the kitchen back there. I don't know. Anyway, so a lot of the stuff we have is just things like hats and so forth, but we have other stuff and we have to be careful about that stuff. All right, so other collaborations. We have various collaborations. We have collaborations with the New York Botanical Garden, the Rochester Academy of Sciences, Buffalo Museum, Britt in Texas, where we're doing a project with an extended specimen network that would be plants collected in the Philippines, and then we have images. Images of those that are connected directly to the plant. You should have the specimen and images taken of that exact plant. And how many are there? Because thousands.
Going to be like 20,000.
20,000. There you go. That's pretty good. So we have various kinds of connections there. I want to highlight one of them, David Warrior, who's in the audience here. Stand up, David. David is in the audience, and he works quite a bit in the herbarium. And so he's produced a catalog of the vascular plants of New York, and you're going well beyond that, right? You're working on a bigger project here. But David is a very important part of our herbarium. It's great to get free work. All right. So then we also have connections to the Cornell Botanical Gardens. Of course, you know, we're always involved with Judy's Day. And this is, we also have been documenting over the years the collections in the botanical gardens. So a lot of the specimens that are grown in the botanical gardens, we have herbarium specimens of those. And you can see over time what we have, because things disappear and so forth. We also have a collaborative project with Towns Peterson, which I don't have the name up here, sorry about that, an NSF project that we're involved with that is documenting African plants. So they're digitizing all of the African specimens. And Zach, I want to point out, Zaki Azedin, who is an undergraduate here, is just finished his PhD. He's going to Harvard, and he was involved in our capturing of the images for the African stuff. He was very interested in the African flora. We also have a lot of undergraduate students that are employed since, let's see, we've had about 70 students employed in the herbarium over the past few years. And these students do various things from imaging to collecting data and putting it into the database, so forth, but a lot of other things as well. We also have a connection to courses. So this was, I guess, when Joyce was teaching the class. Those students usually make collections and they interact with the herbarium. We also have herbarium tours. If you go to the open house, you're going to get a sort of a tour, but it won't be a formal tour, but all the stuff we normally have out on the tour should be there. That center picture is, this is how it can look sometimes. Courses come to have tours, staff comes to have tours, and then we oftentimes, and not that often, but sometimes we have tours from people outside of Cornell that come. There we go. We also have an active group of volunteers. Okay, those are just some of them. And volunteering helps us quite a bit. But you know, it really does a lot for the people who volunteer. Many of these people are retired, and it's a really great thing for them to come in, and they'll oftentimes sit in groups and they're working on the database or imaging or something like that. That's River there, imaging. I'm not going to too much, not enough time. We also have an internship. We started the internship a few years ago, but we had a hiatus because of COVID. And then now that we got money from Tara Atluri, some of you know who Tara was, and from her mother. And so now it's a funded internship. This was the first year of the funded Atluri internship, a summer program. And these are the participants. And of course, there's Justin over there, who is our TA. Now they're already doing the extended specimen stuff in the internship. This is we had them collect. They collected about 500 plant specimens over the summer, but some of what they collected was in the conservatory. So this is the Brownia. Some of you may know that. So we have a specimen of the Brownia and also images of the Brownia. Those will be connected when we put everything up. We can't forget the Varsity Botany Club. Okay, so the Varsity Botany Club was started, what, last year? Have the date? No. Last year. Okay, the Varsity Botany Club came out of basically the students who went to Patagonia kind of got together, and I suggested that they have a club. There is the Hortus Forum, but the Hortus Forum is not about wild botany, collecting plant specimens and all that stuff. So it complements the Hortus Forum. It's not competing with it. Is it, Jonathan? You guys have fights or anything? No. Okay.
Not yet.
Not yet. Okay. So this is the slide they put together, all the kinds of things they do. They've had several lectures already. I think Alejandro gave a lecture. You've had others as well. And then they've had little workshops and things like that. So it's really a great thing. Yeah, we're doing okay. So another program that's kind of associated with the herbarium is the Patagonia program. That's also associated with the Gandolfo lab, because Alejandro works on fossils in Patagonia. I've done various things in Patagonia over the years in terms of research, mostly with Nothofagus, the southern beach. So we had this connection, and a few years ago, Dean Henry came to Bill and said, can anybody teach a class that gets the students involved in hands-on botany? And so I said, well, I've been planning on teaching a vegetation class. Anyway, out of that grew the Patagonia field trip. And then Mrs. Lynch, who's got lots of money, gave us money to do this. And it's been recurrent. We've now been doing it. I think this is the 12th time we'll be taking students this January. So this is a typical route that we might take. We sometimes go into Chile, other times not. In the past few years, we've always ended up down here at the huge glacier, the Perito Moreno, which is a life-changing experience. Here it is up there for a lot of people who've never seen a massive glacier close. It's pretty amazing. So now we have other collections in the herbarium. Over the years, I have kind of rescued things, and then we also developed an image collection. So I'm going to talk a little bit about those things. Kupak, which I'll talk about in a moment, Cornell University Plant Anatomy Collection is a collection of anatomical slides. Cubic is a collection of live plant images mostly. The Horticultural Catalog Collection predates me and is a collection of seed and nursery catalogs, many of which are really beautiful, and you'll be able to see those, some of those, if you come to the open house. We also have the Cornell University Paleobotanical Collection. And 3 or 4 years ago, I kind of negotiated with Bill and Alejandra, said, let's just pull this under the umbrella of the herbarium. Makes it more secure. We can deal with it in a better way. So these things are all administratively now at a certain level under the herbarium, although the CUPC collection clearly will have a separate curator. The conservatory, I'm not involved at all with maintenance or growing of plants, but I and the herbarium accounts have to do with replenishing and deciding on what plants we have in the conservatory for research or teaching. So you could come to me as well as Paul, who's in the back there, if you have any ideas about some plants you'd like to see in the conservatory. But again, the conservatory is not about horticulture per se, or it's a teaching and biodiversity collection, really. We also have a lot of archival materials, and we have, as I said, ethnobotanical artifacts. So the CUPAC, the Cornell University Plant Anatomy Collection, came after I came in 1985, and over the years I'd see, well, there's these boxes of slides and anatomical slides, and nobody wants them. So I started collecting those things together. We now have a massive collection, and Maria Gandolfo Alejandra is the curator. We have thousands of online searchable images. I'll give you a demo of that in a couple minutes. What time do we officially end? 5 after?
Yeah, 10 after.
10 after. Okay, we're fine. Okay. Now here's a little bit of information about CUPAC. We have a lot of really important plant anatomists from Cornell and a few from other places where we have slides. Many of these slides have to do with their published work, and we try to connect those up. You might call that an extended specimen. They're vouchers, in other words. So maybe Eames did a study of some aspect of anatomy. We have the slides, and we also may have a herbarium specimen that goes with it. We're still in the process of connecting all this stuff up. Those are some of the people that have important collections. Look, Charlie Uhlder, we've got 14,000 of his slides. Now, of course, many of these slides in these cases are sequential serial sections. So we'll pick out of those, but the slides themselves, we'll digitize only some of them, but the slides themselves will be available if somebody wants to look at the whole series. Let's go on to this, the cubic. This grew out of the fusion of 2 different plant image databases, one of which I started, which is called plantsystematics.org. So I wrote some software to manage images and MySQL databases and do a web interface. And then Dan Nickrent contacted me. I put a little thing on the site if you're interested in this. So Dan contacted me, around 2003 or something, somewhere in that area. And so I set him up. He started his own site called Phyto Images using the same software. And then recently we decided that we're going to fuse these together. Now, these were never part of the Cornell system. So now they are. That's the whole point. Cubic denotes that they're coming into the Cornell system because Dan and I aren't going to be here forever. So they have to have some continuity going forward. We now have about 220,000 images in that database. Now, the Cubic is a really important resource for images. Unlike something like iNaturalist, this is highly curated in the sense that it's not just a bunch of pictures that people take. These are oftentimes taken with particular reasons in mind, and I'll show you. There we're trying to get the characteristics of the plants. All right. Advanced search functions, so forth, so on. We won't worry too much about that. Oh, here it is. 83,000 images in this project with Brit. And that's a big project. All right. So Dan uses our collection almost exclusively to develop materials for his class. These images all came out of our database. Is that correct, Dan? Yes. Okay. So you can see the usefulness. And again, unlike iNaturalist, I'm not dissing iNaturalist, don't worry. But these are the kinds of images we often find, images that actually are zooming in and have detail And they're keyworded, most of them, and we're adding keywords so that you can find what you want out of it. We also have the catalog collection, which I mentioned. We have 135,000 catalogs, and you'll get to see these. In fact, I think this one might be on the table up there right now. But these, there was a certain period of time when these were like works of art, and very nice. But ours go back to the 1790s. And here it is. This is in the vault downstairs. Some of it's in the vault. A lot of it's in the vault. Some of it's still up in the herbarium. And truthfully, we haven't figured out exactly what to do with this. It's hard to get money for this because it's horticultural and it's not growing plants. Years ago, I tried to get some grants from horticultural societies, and they didn't care about the taxonomy of horticultural plants and things like that. Now, we also, COVID really generated this for us. When COVID hit, I had to send Anna and Peter home, and we didn't exactly have a lot that they could do there. So I set it up so that they could start to populate our biography database. So we now have a biography database of about, well, in the database, we have 3,780 people. Some of you are probably in there and don't know it, because we only bring the biographies public after the person passes. So the point is that we have 2002 that have passed, more or less. There's more now. But this is probably one of the best places you can find information about female botanists, women botanists. And then I've categorized these things, and you can search. You can get a list of plant anatomists, of Bailey Hortorium affiliated biographies, Cornell affiliated biographies. We have 167 paleobotanists, 46 cytogeneticists, and so forth. CUPC, I'm not going to talk much about. This is the paleo collection. This was a lot. Who started it actually, Bill?
Well, Banks certainly was.
Banks was the big guy, but I thought there was some other stuff there before that.
Yeah, the small teaching collection.
Small teaching collection. All right. So we just started to deal with this. There's a lot of stuff. It's difficult. Okay. So that's for the future. Bill is spearheading, writing a grant to get an improvement, like new cases and digitization, things like that. So now I want to go into the little demo here. Oh God, I gotta find the mouse. There it is. Wow. Okay, everybody close their eyes.
We can't see it.
I know that was a joke. All right. So I'm just going to take you now. I want to point out this will be the it shouldn't be doing that, but this is the new website. It's not online yet, so you're getting a preview, and you can see this would be the herbarium.
We haven't, you have to drag it over.
Where do I drag it
So
to?
drag it from that screen over to this screen. Like actually physically,
Okay.
well, not physically.
Well, it's okay. I can just talk about it. You guys don't need to see
There
it.
you go.
You guys don't need to see it, do you? All right. Can I get it centered there? So
That's good.
that's good enough.
Screen's
All right.
So
All right. So this is, yeah, it's all backwards upside down. So this would be the main herbarium site. This will be cubic. And if we scroll down, see, no, now I can see it up there. Maybe. Oh, wow, I've got a mouse up there too. All right, there's CUPAC, CUPC, Conservatory, the catalog. We also, which I haven't mentioned, we have a huge literature database. Well, not huge relative to Google. But during COVID as well, I had to have things for Anna and Peter to do. So we started to capture old literature. So I could show you an example of this. We have about 220,000 items in our literature database. We have Botanical Gazette, Old American Journal of Botany, Evolution, Ecology, all those things. I won't tell you how I got all that stuff. I could tell you. Okay, so at any rate, Then we have a bunch of other stuff, but let's go back up. Rotating. Yeah, it's down there. Oh, we don't have this populated yet. Well, I'll just show you VH Bios quickly first. Okay, here's VH Bios. This is public. If you want to go there, you can go to it. There is the URL up there if you want to write it down. And today, we have born on this date, which it gives you if you go to the site. My favorite guy, Lysenko, was born today. And Francis Marion Ownby, isn't he your grandfather or something? No. Who's Heiser? Who did Heiser?
Baker.
Baker, so it wasn't Ownby. I thought it was. Okay, so Ownby was really important in polyploid studies and so forth. Lysenko, we're going back to Lysenko soon after the next national election. So at any rate, but you can see, for instance, we have women in botany here. You can click on that. I think we have 406 women in botany here. And I think it's relatively complete. The way we did this is we started by taking our database of specimens and then making biographies, entries on everybody who had collected. So we have a lot of people here that you won't find in other places. And then we continue to add to it, all right? So if you click on one of these, you can get more information. It varies quite a bit. And if any students or anybody would, here's the problem we have. Every time we hire a student, I say, are you interested in history? Do you want to work on the biographies? SIP. The students are not interested in history or biographies or anything like that at this point. Okay, but for instance, let's go down, see if there's anybody. There is. Okay. You guys can visit that as you see fit. But it's extremely useful. We also, now again, we don't have living people in this. We have plant anatomists, so forth and so on. So now let's go back. And I can show you, if we go back up, right? Well, let's just go to Kupak. So this, Kupak has not been updated. This is a public site. This is not the public site URL, but you can find it pretty easily. All right. So this will be the new one. Now I want to point out, this is not the launch thing. So we're going to putting backgrounds and colors on this. You're seeing the basic thing that has functionality. Whoa, that's nice. I remember that. Okay, so then we come here, so we can search, and you guys could yell out something. These are anatomical features. So if I do this, did I misspell it?
Yeah.
No, I didn't. Okay, so sieve cells.
It's just that,
I don't know who put that in there. Right. That's not what I wanted to show you. So let's go back.
Must be the government.
We can fix that. What do you want to search? Trachea or something like that? So what I do want to show you though is that you can measure. Pardon?
Oh, no.
Tricho.
Well, that's not anatomy.
Well, wouldn't it be in there though?
It'll be in there. Let's do trachea. All right, so here's a whole bunch of tracheas. We click on here, we can actually measure images here. Now, one of the problems is I'm going to make it in multiple colors. It's red, so there we go. That didn't work because you didn't see it. See, it's no good on red. I'm going to make it so that the measurement lines are different colors. Here's a green. Let's look at that. You'll see it more easily. But I have to go to the other one. Image, measure image. All right, so you can come in here and you can measure these things. So you see, so that's 142 micrometers. You can save the measurements up here. You click on that. All right, so they come up here. I also have a setting that's not for the public that I can load it into a database directly. So if you want to make measurements. We're getting ready to do this, and I'll show you with the plant specimens, our herbarium specimens. We'll come back here. This is hard, guys, sorry. So if we come back up here, you can see that we have searchable database here. So of course, we'll put in my favorite genus here. Now, again, all of this is not ready to go public yet, but it will be very soon. One of the things that I've really made an effort to do is make it fast. That's the first time that was loaded on this computer, so it's not cached or anything. All right. So you see that it's very fast. And then it also, I mean, I've looked at a lot of the online specimen databases and I find it to be a little confusing. This is very simple and straightforward. But if we want to measure a specimen, you can come here. Now, this screen is not very good, but you can say, let's say you wanted to measure the width of that leaf over there. Again, it's the same thing. That's 9 centimeters. And then I can load that up. And so if you're doing it not through my database, you can still just paste your values. Okay. So let's go back. We're about done. Yes. Perfect. So we want to go to cubic. Cubic is very impressive. And I have to give Dan and our other collaborators a lot of credit on this. It's myself, Dan, Peter Pelser, Mihai Costea, is that how you said? And Julie Barcelona, various other people have been involved in this. So we have 232,000 images. So this is the one I like to do because it's so impressive. You guys familiar with Rubiaceae? Oh, look how I spelled that.
Sorry, no.
It's already, okay. Caps lock's on, all right. I think it'll still work, we'll find out. So then I can search stipules, right? No, it doesn't like it.
It's an E.
Yeah, it is.
No, it doesn't need an E.
Oh.
Sorry, guys. What it needs is to not have, it must be case sensitive. So we're going to come back here. And hopefully, you never know, there could be a glitch here, but I hope not. Rubiaceae stipule. There you go. See how quick it loads? Okay, so that actually has 372 images of Rubiaceae stipules, right? That's just one of the pages there. And see, so we're making an effort to make comparable images across the data, our image set here. And that's it. I've got to quit now. So, oh no, I don't have to quit. I have to go to the next slide. And so what's the neo-herbarium? Okay, so we have a new paradigm that extends the concept of specimen. That I think I hopefully I made that clear. And not only in terms of the actual specimen, but the metadata that's involved. Combines links to multiple aspects of plant biology in a digital framework. It recognizes the value of the neo-herbarium. Of the herbarium as a global archive of genotype, phenotype data. There are going to be undoubtedly new and unanticipated uses as well. And I think I can't emphasize that the herbarium has become a hub for undergraduate students, right, Claire? Yeah.
Yes.
The undergraduate students are always in and out of there, and it became the center for the club as well. So adapting herbarium practices to new uses. I forgot to put, perhaps we should always try to get silica samples. But one of the things is just to dry without heat or minimal heat to try to get maybe a specimen plus a population sample, DNA conscious collecting and databasing, labeling in the field using, I already have software that does this, but it's not quite ready to go out. And then we're also going to propose that the herbarium could be a service for any of you guys who don't really care to press your plants. Let's say you do an experiment, you want to document it. You can bring the plants to us, and then we can press them, put them in the herbarium, and they'll be there for 200 years, 500 years. But you can then cite those specimens. If anybody has a question about what you were working on, we'll be right there. Thank you. Probably a little lower. Any questions? No question. Brandy?
It's a subject that either facility or any other that say
Cross sections of trees?
data.
I've never seen that. But you can do anything you can imagine.
Yeah, because that it'd be a way to calibrate like.
Well, the other thing is if you document and say have the tree, maybe you could look at isotopes later, you get population samples and so forth. The main thing though, I think that we have already are these small population samples that nobody, as far as I know. And so you can look at, what's the variation? We've got 10 plants from a site from 1925 and the thing's now extinct. Was it, inbred or whatever? So, yes, that's it. Oh,
Why are herbaria have such hard times?
because of DNA. No, I mean, it's space. Every square foot costs something. Every person you hire costs, right? And so since the trend has been to go more to molecular studies, I think herbaria has been viewed as not that important to the general trend in plant biology and other areas. I'm trying to make the point here that that doesn't need to be. It shouldn't. Does that make sense? It's all a cost benefit, And the other thing is what happens is if there's a hire, sometimes, there was a herbarium curator, and then when they hire somebody who has nothing to do with it, send the herbarium off somewhere, right? That happens as well.
Following up on the sending herbarium off, based on your experience, you already touched on some of this in the talk. What is the value of keeping herbaria, especially at academic institutions? Because we could send it to NYBG, we could send it to Bobot, but we have this undergraduate population and that opportunity for exposure. But they can.
Well, first for
Yeah, absolutely.
Okay. Undergraduate participation, teaching experience is a huge thing. Now, does that justify having a 950,000 specimen variant? Probably not. But I also think that most people who are doing research, even now, could benefit from documenting their specimens, depositing the variant. It's a permanent record. That's what science is about, right? Repeatability. So if you do a study and you just say this was Thalictrum whatever, right? If you say that's what it was, everybody has to take your word for it. If you make a voucher, then somebody can go back and check it. That's one of the major uses of herbarium. Why they shouldn't all be in one place is probably a matter of administration and nuclear war. It's better to have them dispersed. You laugh at that, but C.H. Muller, we got the oak collection from, he actually thought very carefully about it before he gave it to us at Cornell. He's really old. He said, I've considered it, and I think Cornell is not going to be likely to be hit. He didn't want his herbarium to be in New York City.
Yeah.
That one. Yeah. All right. Okay, thank you.
This has been a production of Cornell University, on the web at cornell.edu.